So on the left here, you'll see an outline of the Luckiamute River Watershed, and on the right, you're going to see an outline of what we're going to go over tonight. So start with looking at some the motivations behind our monitoring program and why we even go through all the work of collecting data around our watersheds. Like why that's a lot of time and effort. Why are we even going to do that? Then we're going to go over a bit of how we monitor, the methods we use, the kind of data we collect. And then we're going to end talking about how we use the data, how we inform our restoration projects with this kind of data. So hopefully you'll kind of learn a bit about the behind the scenes of our restoration work in the watershed. But what I really hope you kind of come away with tonight is a better understanding of the complexities in our natural systems, right? A lot of times when people talk about collecting data out in nature, it ends up sounding like, oh, we're going to go out there and try to understand everything that's going on out there. But these systems are so complex that we can't do that. That's almost impossible. Especially for an organization our size, we do not have the resources to do that. But we're trying to get a better understanding, or at least understand the aspects of these natural systems that we closely interact with and are most useful for us in our restoration work. So the number one most important thing about our monitoring program is that all of our monitoring is in direct service of our restoration work. A lot of other people, and like a lot of research institutions who do monitoring and data collection out in natural systems, their goal might be to try to increase understanding of these natural systems, or try to build humanity's total knowledge set. But for us, that's not what the business we're involved in. We are informing our restoration work. And we do our restoration work for a few reasons. These are kind of my summarizing of our reasons, so that's not the Watershed Council's wording, but I like to think of it as, we do restoration work in order to conserve natural processes, protect ecosystem services, and support endangered and native species. Now, those are some technical words, so I'm going to explain those a little bit. We conserve natural processes. A lot of people kind of think about nature as the things that are out there in nature. But I think it's a little more useful when we're trying to talk about interacting with nature and natural systems, is thinking about the processes that are going on out there. So some examples you might think of are like the water cycle, the carbon cycle, how our river systems develop and flow down through our watershed, how forests develop in successional stages. All of those are the processes that go on out there. And if we can understand those more, we can interact with them and influence what is out there more effectively. We're also doing restoration to protect ecosystem services. A lot of times when we think about restoration work, we kind of tunnel vision on this environmentalist side of things, right? Trying to look at restore nature for the sake of nature. But ecosystem services are things that the ecosystems provide that are of direct value to humans. And those are very important parts of restoration as well. Having clean water and clean air is valuable for all of us. We could even probably put a monetary value on that service. Having productive forests to gather resources from, and having amazing recreational areas. A lot of our streams are beautiful and many people like to recreate in them, and that is of value to humanity. So while we may not do restoration primarily targeting those, restoration has a major impact on those services. That's important not to ignore. And then finally, I think the thing that's pretty clear for everyone is we do restoration to support native and threatened species. We tend to focus our restoration work on salmon and steelhead because they are kind of pseudo-indicator species. An indicator species is a species of organism that we can look at, and if we can assess its health, it's a good metric to look at the ecosystem as a whole. So if salmon and steelhead are doing well in our streams and are thriving, then we can say that the streams as a whole are probably doing pretty well also. So we focus a lot of our work working with salmon and steelhead. And so kind of big picture summary of why we monitor. I'm going to go through a little brief flow chart with you in my head of like, why do we need to monitor? Why can't we just do this restoration work without monitoring? I mean, we see streams out there. We see areas that could very well benefit from restoration work. Why do we actually go through all the effort of collecting this data, doing these analyses, all that kind of stuff? Because it's a lot of effort. But there's some important reasons. So here's kind of our thought process. So first of all, we target salmon and steelhead as our species we're looking to help. Salmon and steelhead require cold, clear water to spawn and appropriate spawning gravel in these streams for them to thrive. They just need that. You can think of it as like a cozy maternity ward, except for it's freezing cold and there's way more gravel involved. So yeah, that's kind of what we're aiming for. In our watersheds, temperature is frequently the limiting factor, preventing more of the watershed from being high quality habitat. So that's kind of the thing that we can target to increase habitat. And we can do restoration work to improve the temperature in a lot of our streams, or to at least limit any warming conditions. But we need to know where and when those temperatures are occurring around our watershed in order to appropriately target our restoration work. And monitoring gives us that information to be able to make those decisions. So, if we kind of work backwards through it, we monitor in our watershed to gather the data to be able to apply our restoration projects to address temperatures in our streams that are limiting salmon and steelhead habitat. So that's the big picture reason behind our monitoring. Yeah, I'm going to go into more information about the details of that here in a little bit. But I want to take a little brief tangent to talk a little bit about kind of the blessing of the water quality that we have in this area, right? We are pretty lucky to be living in Western Oregon, and our watersheds, especially compared to most of the US and a lot of the world, we have incredibly high water quality for how close our human population centers are to the water sources. It's a real big blessing. We gain lots of things from it. We don't have to spend as much energy treating our water for population centers. That's a big deal. We have, amazing recreation opportunities. I'm sure many of you have gone up into the hills and jumped in a creek and not worried about getting sick at all from just jumping in the water. Many places in the world can't say that. And my personal favorite is I love just drinking my tap water and not worrying about the flavor at all. I really enjoy that. But this really high water quality comes with pretty big responsibilities, right? Our natural systems developed under conditions of high water quality. So they aren't particularly resilient to massive changes in that water quality. And since a lot of our human developed areas in this part of the Willamette Valley are very close to streams, and we interact with the streams all the time. A lot of our roads go right next to it. Our streams aren't just way out in the wilderness untouched by humans. We are around them all the time. And so there's lots of potential for human issues in our streams. So we have a major blessing from our high water quality, but we also need to protect that. We talked about how we need to monitor to get the data for our restoration work. Okay, so we got that. But why do we have to do that monitoring, right? There's lots of governmental organizations and other research organizations that are collecting data in our watershed. Why can't we just use their data? There's some problems with that. We can go back to the beginning of monitoring work in the Luckiamute Watershed and in the Marys River Watershed. That kind of began around the 1910s with the USGS river gauging stations. Those were about the first continuous monitoring work done in the watersheds. It's really long-running data, but there's only one station on the Luckiamute River and one station on the Marys River, and they're really focused on water volume in the streams for agricultural water use and to manage the reservoir supplies. So it doesn't really apply for what our restoration work we're looking at. The EPA tracks pollution sources, in our watersheds, but they only really monitor fourth order and larger streams, so kind of streams that are too big for what we are looking at. The Forest Service and BLM, they do lots of monitoring, but they're restricted to federal property for their monitoring. Researchers at OSU, U of O, and WU are often looking at similar kind of questions to what we're looking at, but they have specific project areas for specific project periods, so we can't just rely on their data for all of our work. A lot of the municipalities and counties are monitoring, but they are often looking at city water supply and water treatment outflows. So really, the only organization that's collecting data that we could use to inform our restoration work would be the Oregon Department of Fish and Wildlife and their Aquatic Inventories Program. It's a pretty amazing data set. It covers basically all of the state, and it kind of quantifies and qualifies the amount of freshwater habitat in our streams. It's pretty amazing, but there's some problems with it. It isn't updated yearly, and it doesn't really have the resolution to look at each of our streams and each part of our streams and be able to understand the temperature patterns in those areas. So it's not quite sufficient to answer our questions. So basically, we need to collect our own data because it's not out there from other people collecting it, which is fine. That's why we have our monitoring program. So it all works out. So we need to collect our data. What do we actually do when collecting that data? The first part, if you're ever collecting data in a natural setting, is you have to carefully define the questions you're trying to answer. Because, if you go out in our watersheds, immediately there's thousands of questions like what's going on out here? What is it interacting with each other? How are all of these different all these plants and animals, are they thriving or are they not? What are the challenges they're facing? There's a bazillion questions, including like, do fish prefer like indie rock or smooth jazz when they're getting down to their spawning? Like, what's going on out there? We don't know. So we have to limit our questions to, to fit in our funding sources. So both, the Marys River and the Luckiamute Watershed Councils have slightly different monitoring questions, but they kind of summarize to something like this. Where do stream temperatures exceed spawning and rearing criteria in our watersheds? So what parts of, what streams in our watersheds are nice and cool, high quality habitat from a temperature perspective? Which areas are too warm, they exceed the thresholds that, that fish need? And what areas are kind of in the middle, iffy conditions that we can kind of work with and try to influence? And our second kind of big question is, what is the distribution of key species around our watershed? So, we mostly look at salmon and steelhead, where are they spawning in our watershed? But there's also other species like freshwater mussels, Pacific lamprey, western pond turtle, and even some like not necessarily threatened ones like beavers, where they are located in our watershed plays a big role in our restoration work. So understanding that's very key. And then if we can answer both these questions, we can kind of compare their results, overlap them and see which areas are high quality habitat and are animals using them, which areas are animals using that aren't high quality habitats, and can we explain why that's happening? This can help drive our restoration work. And to answer these two questions, the Oregon Department of Fish and Wildlife Aquatic Inventories Program data just isn't sufficient. We need to go beyond that to collect our own stuff. So we have to collect our own data, and that kind of comes in two forms. These maps on the left are very detailed. I'm not expecting you to look at what's on there. All I want you to see is that this left map shows all of our temperature monitoring locations around the two watersheds. The Luckiamute's on top and the Marys River is on the bottom. And then this kind of map in the middle shows all of our locations where we take eDNA samples around the watershed. I'm going to explain both of these here in a second. So our temperature monitoring program, every summer we go out and deploy a whole bunch of temperature monitoring sensors that look like this. So it's pretty small. This is a self-contained little computer that collects and records all the data we need. So we set these up, we place them in the rivers, and every half an hour it reads the temperature of the stream and records it to its little database. At the end of the summer, we walk around, collect all of them, and then we have a pretty big data set from that past summer. We collect data only in the summer months because, as you all know in this part of Oregon, in the summer, summers are much warmer than the rest of the year, and they're much drier. So our rivers really only hit peak temperature in the summer, and they're never even getting close in any other time of the year. So to save resources and not be monitoring all year round for no particular reason, we only monitor in the summer, usually from June to October is when we kind of focus. So that's pretty simple. We just collect stream temperature, but our eDNA work is a little more interesting, so I want to explain a bit more of that. So you probably have all heard of DNA, deoxyribonucleic acid. It's like the genetic blueprint of life. All life has it. But what is eDNA? So the E in eDNA stands for environmental. So environmental DNA is not DNA we gather from a particular species. It's DNA we find in the environment. So you could kind of compare it like this. So regular DNA sampling, you would say like, oh, here's a fish. I found a fish. I'm going to take a little bit of it, the fish, back to the lab and see what its DNA looks like. Okay, that's pretty typical. But eDNA is kind of the reverse process of that. We go out into the environment, we just take a sample from the environment, and then we send that to a lab, and we see, hey, do you find any coho salmon, any steelhead, any western pearl shell mussel DNA in this sample? And if you do, then we can say, hey, they must be present in the environment because we found their DNA there. So it's kind of a, it's a more efficient and more like an introvert-friendly way of sampling. You don't actually have to go up and find that species that you're looking for. You can just kind of say, hey, were they in the area? We can kind of detect their traces. So it helps a lot, and it's a method that's only really been developed in the past decade or so to be really feasible and like within the price point that we could actually afford it as part of our monitoring. And it kind of transforms the kind of questions we can look at, because besides eDNA monitoring, the only way to get information about whether or not a particular species is present in a stream is to pay someone to go out in the stream and look for the species with their own eyes. Like, that's the only other way to do that. So that is way more expensive than going out and taking a little sample and sending it to a lab. So it kind of opens up this possibility of questions we can answer. As you can see on this map, we can take samples all over the watershed, and it's not really that expensive, and we can get data from all over the place. Yeah, so those are our two main pillars of our monitoring work. We do both of these each summer. And these monitoring plans of all these positions, we are always adapting them and changing them to better answer our questions. A great example is that in the Luckiamute Watershed Council, you can see a lot of our monitoring points are kind of up in the headwaters, away from the valley bottom on the right there. And that's because we often have been focusing on the headwater streams to see that's where most of the salmon are spawning. So let's look at where the salmon are spawning and understand more about that. But in recent years, in the Marys River Watershed, we've been adding temperature probes in these lower streams down in the agricultural parts of the watershed, which the salmon aren't spawning there. But recent academic research has been showing that salmon in different parts of their life stage use different parts of the river system. So while they may, lay their eggs and the little tiny fish will develop in the headwaters, as they grow and develop, they go to other parts of the watershed. And so only focusing on just the headwaters kind of leaves off a big chunk of the fish's life cycle, where they are trying to fend in unrestored streams. So we're probably going to try to do something like that similarly in the Luckiamute pretty soon, try to get some more information about these agricultural areas. And you can kind of see those yellow dots at the top there. I don't know, it might be too small, but we're kind of moving. We're going to add some sensors in the Teal Creek and Little Luckiamute parts of the watershed this coming summer. So we're kind of always expanding and growing to better answer our restoration needs. Another part of our restoration work that is really important to mention is the very high involvement of volunteers and landowners in our monitoring program. This is something that makes our program kind of special and really powerful, is that, I'm the monitoring person and I can't do all of this myself. Like, that just doesn't work. So we have lots of volunteer help through both watershed councils who go out and help with all the data collection, all of that kind of side of things, as well as the data analysis. And I'm still just one person, and so I only have one opinion on what we should do, and it's not always the best opinion. So having other minds who help, advise on, hey, you should add stations over there, or maybe you should try this analysis and see what that tells you. That's very valuable. So all the volunteers who collect data and help with our data analysis, it's all very valuable. And then a really powerful part of our monitoring program is the landowner participation. As I was talking about all the other monitoring agencies earlier, you probably noticed that a lot of them are the government. The government collects a lot of data, and that kind of makes sense. But the government has limited bandwidth and cannot work with many local landowners all at the same time. It's really difficult for the government to do that. But as a local group of watershed people, that's much easier for us to do. And so a big part of our monitoring work is reaching out to local landowners and working with them to gain access to their land to be able to monitor on their property. This kind of opens up a lot of the watershed to us to monitor that many other groups wouldn't be able to monitor. So it allows us access to very valuable locations, but it's also beneficial for the other people, for the landowners involved in the program, because they get access to people who, for free, from their point of view, are like researching their property and seeing what the patterns are in their streams and helping inform their land management decisions. So it's a very win-win for everyone involved. Between both watershed councils, we have over 30 landowners and their families who participate in this work. So it ends up being a really important part of our work specifically. So between both of these sides of our monitoring program, between all the volunteers and all the landowners, we collect a lot of data. But what do we actually do with that data once we got it? We got to make good use of it because we put a lot of effort into collecting it. So if you ever look at environmental data, you'll probably recognize similar things in these plots. I'm not going to go through these in very much detail, so don't worry about the specifics. But we can plot it. This is just one summers of data. This is for the Marys River. It's all the stations on the main stem Marys River going from upstream all the way down to the bottom. So it just kind of warms as you go along. So you can do stuff like this. You can take one location on the river and have all the years together and look at the differences between different years. You can kind of do fancy stuff with histograms and look at like the percentage of days that are above a certain threshold of temperature. All that's, it's fine and interesting and stuff. And we do that every year. We do an analysis of that kind of stuff every year for all the new data we have. And it's available, so you can go look around, or you can ask me, the monitoring coordinator, if you want to take a look at that kind of stuff we have. But tonight, I want to focus more on how we use these results in terms of informing our restoration work, because I think that's a little bit more interesting. And I'm going to look at that at 3 different scales. So the first scale is kind of local, project-specific. So, at one stream, one restoration project we're doing there, how do we use our monitoring data to improve that project? And then I'm going to talk a little bit about kind of multi-stream watershed scale. How do we look at all the streams in context, and how do we prioritize projects across our watershed? And then finally, I want to talk a little bit about some kind of special kinds of analyses and understandings that we can come to when we have sufficient data. In the Luckiamute River Watershed, this coming summer will be the 10th year of continuous monitoring at some of our stations, which is a lot of data. Getting a decade of data is kind of a big deal. And it opens up new analyses that you simply couldn't do with less data. Some of these smaller scale analyses you could do with one summer's worth of data. It would be more accurate if you get more years, and you can be more confident in your results with more years. But when you have almost a decade of data, you can start to do things you simply couldn't do with less information. So I want to go through a little bit of that. So we're going to start with project-specific uses. I'm going to use this project as an example. This project, this map on the right, is of North Fork PD. It's a project currently in development. We haven't done the restoration yet, but we have money that we're planning the project. And it's a great example of how we use our monitoring data to help inform our project development. So when you think about the current temperature in a stream at a specific time, there are a lot of factors that go into determining what that temperature of the water is. There's a lot of warming factors. So some things we commonly consider are like solar radiation, the sunlight shining on the stream, it warms up the water. There's also the air temperature around the stream. If the air above the stream is warmer than the water itself, it will be warming the water. There's lots of groundwater inflow from the surrounding hill slopes into a stream. And whether that groundwater is warmer or colder than the stream water, it could warm or cool the stream. There's kind of a unique phenomenon in our kind of streams called hypereic flow. And that is the flow in between all of the rocks in the stream bed. It's actually much cooler down in the stream bed. So as stream water flows in and out of those rocks, it cools down. As it comes back up to the main stream channel, it ends up cooling the stream. So a stream that has lots of gravel in its bed is going to actually be quite a bit cooler than a stream that's just on bedrock. So that's kind of interesting. But then also just the temperature of the water upstream matters a lot, right? That's kind of like the base that's the base temperature that all these other factors are going to influence. So there's all these different factors we can think about and how they influence the stream temperature at a given point, at a given time. And we can do restoration work that addresses different ones of these warming factors. So for some examples, if we plant trees and shrubs right along the riverbank, and as they grow up, they grow over and shade the stream, that limits the amount of solar radiation, the sunlight, hitting the stream, and so it reduces the amount of warming that way. If we also plant trees and other vegetation in the surrounding area around the stream, that's going to lower the overall air temperature in the area and reduce warming that way as well. And even if we do something like putting log structures in, where we kind of place logs in a stream, kind of in a partial blockage, so it slows down the stream flow. When you do that, when the stream flow slows down, it drops a lot of the gravel and sediment it was carrying with it as it flows downstream. And as those accumulate above the log structure, that deepens the amount of gravel in the stream bed and increases the amount of hypereic flow that's flowing through that gravel. So you get some cooling that way as well. So we have all these different projects we can implement, and they affect the cooling and the warming of streams in different ways. And so that helps inform. If we know about the temperature dynamics of a stream, we can use that in our restoration project planning. Another aspect of restoration project planning on this scale is where exactly we're going to place the project, right? So, we can say, hey, we want to do a project here on North Fork PD, somewhere in here. But where do we actually put the project? Are we going to put it way up at the top? We're going to put it at the bottom? We're going to put it in the middle? Or we're going to try to do it along the whole length of it? We're going to concentrate in a specific area? There's lots of choices that go into that. And our temperature data helps a lot with making that decision. On this map, it's kind of like an abstract of the warming that's going on in this, in this system. It's a little hard to see in this lighting, but you can see this kind of the lines that represent the streams are different colors. And up at the top, they're nice and blue. They're around 16.5 degrees Celsius at the peak temperature in the summer. But then as it goes down, it turns purple and pink and red indicates around 18 degrees Celsius at the peak time of the summer. And that's right about the threshold of being, it's just about too warm for fish at that point. So we can see the transition of temperature through our project areas, and we can place our restoration work at areas where it's going to limit the most warming. So we can see up at this top area, it goes from blue to red really quickly. There's a lot of warming there. At this lower area, it takes a longer time for it to reach red. And so we can place our project in that direct warming area, and that's going to have the most effect. And as it turns out, that's basically where the North Fork PD project is going to be located. It's right in that area of the most warming, so it's going to have the greatest effect. Yeah, so our temperature data and also our species, where they're located data, helps a lot in kind of planning these projects specifically. So this is a great example. This kind of same work is done in the Marys River Watershed as well. Some great examples are Oak Creek, if you ever visited the Oak Creek Trailhead in the McDonald Forest. There's restoration going on there that had this same kind of analysis done. And Devitt Creek and Shopout Creeks are also really important streams in the Marys watershed. Yeah, so this is very useful, and we can do it for most of our projects where we have temperature data for. When we kind of zoom out to that second scale of restoration work, when we have temperature data for a bunch of streams in our watershed, we can get a better idea of the patterns across the watershed as a whole. So this map is of the Marys River Watershed, and it kind of indicates some areas that are, just to my mind, there's nothing particularly specific about these areas, but to my mind, from looking at the temperature data for a long time, we can identify some areas that are of high importance and high quality stream habitat. And we can do the kind of same sort of thing for the Luckiamute watershed. And so when we have, this kind of information, this kind of understanding of our watersheds, we can use it to prioritize where projects should be developed in our watershed. We can see the areas that have the highest quality stream habitat where we should work to improve. They have high quality habitat from a temperature perspective. They have really good temperatures. But if there's lacking in stream bed gravel, we can do projects to get more of that in those locations. If there's areas with great stream bed habitat but not great temperatures, we can work to address the temperatures there. If there's areas that we know of a lot of warming that are that are adding heat to the whole downstream system, we can look at those areas, try to implement projects there. So this helps us a lot with our internal planning and development of restoration projects. But it also helps us acquire funding for these projects, right? We don't control the money printer, and we can't just do projects all across the watershed everywhere we want to, right? We are very limited by the grant funding we can get to implement our projects. And we have to apply for grants. We have to submit proposals, and they have to get selected from a whole array of other projects. And so if we can submit data alongside our grant proposals showing, look, we're not just trying to convince you that this stream needs restoration, we have data to back it up that shows it needs restoration, that goes a long way in getting our funding for our projects. You may kind of think like we're kind of tricking the system, but it actually helps the granting agencies as well, because they need to make decisions, for the whole state of Oregon, where are they going to put their money? And when they have data from all of these different proposed projects, they can better make a decision and prioritize projects across the state better as well. So, this helps at many different levels, and it's really well worth the investment in our monitoring work. Yeah, and then finally, if we kind of look at the largest scale, so by this, I want to remind you, I want to talk about analyses that require the kind of data we're just getting to, recently, where we have almost a decade of data. We can start to look at more specific causes of specific patterns in our watershed. So those results you see up here now, I'm going to explain them in a second, so don't worry about trying to understand them all on your own. This was an analysis that could only be done with nearly a decade of data. So we're just starting to get to it now. Basically, I modeled the temperature in different regions of our watershed and looked at the relative influence of different other environmental factors on the temperature. So what was the temperature most closely associated with in these sub-watersheds? So we can see up here on the top left, PD, this is the PD Creek area, so it's like the North Fork and South Fork of PD Creek and all the surrounding tribs. They're all kind of averaged into this one pie chart. And we can see the Rittner Creek area down here and the Vincent and Plunkett Creek area up there. And let's start with the Vincent Plunkett Creek area. We can see that the very large segment of it, 58% of the pie, is controlled by the discharge, which just means the amount of water in the stream. That's just a fancy term for water in the stream. We can see in that system, discharge is a major driver of the stream temperature, as well as the green segment, which is air temperature, is also a pretty major aspect. But that's very different from these other two areas, right? In this Rittner Creek area, we can see that discharge is only 16%. It's a pretty minor part of this pie, but solar radiation and air temperature have a much larger impact on the stream temperature. And in the PD Creek area, we can see that solar radiation and discharge have a big effect, but air temperature, while it was 39% of the influence in the Rittner area, is only 3% in the PD area. And so we can see there's actually pretty big differences between parts of our watershed, and if we just treat them all the same, we're not going to get as effective results as we could have if we kind of tailor our restoration work based on the warming causes we're seeing in our watershed. So, based on the explanation of the different effects of different restoration work that we talked about earlier, like planting, reducing solar radiation, and air temperature, reducing warming that way, we can see that the Rittner Creek area could probably benefit from a lot more planting in that area, a lot more vegetation. But in the Vincent Plunkett area, it seems like there's already quite a bit of vegetation. The solar radiation and air temperature aren't really driving any warming there. It's more the amount of water in the stream is driving the warming there. So we can use this across all our streams. And since doing this analysis, I expanded it a little bit farther to include some more kind of static variables. So like the elevation of the stream or the underlying geology of the bedrock in the area. And those also had some minor effects, so it was interesting to see those added in. For the Marys River Watershed, we're not quite at the same near decade of data. They have some gaps in their data, which kind of prevent this from being super accurate. But they're also trying to reach a point where we can do some stuff similar like this in their watershed. So that was kind of the modeling work. Also in this, multi-year analysis kind of stuff, I want to talk a little bit about climate change resilience and how we can think about that in our restoration work. So we saw this plot earlier. This was just one station and all the different data from each year on record at that station. So there's plenty of pretty solid evidence out there showing that in the Willamette Valley, there's going to be pretty significant effects of climate change, especially some warming in our Willamette Valley habitat, in our Willamette Valley stream systems. But it's not necessarily going to be a uniform increase of, everywhere, all the temperatures all the time are going to be 2 degrees warmer or whatever. It's going to come in the form of more extreme warming and drought events, which are going to bump the average up over time, even though day to day it's going to be, not necessarily too different. So how can we address that in our restoration work? Well, when we have a high enough data set, we can look back in time and see years which kind of stand out as anomalous. I don't know if you can see it on this chart, but 2021, this red line here, was much warmer and drier than all the other years in our database. Every year so far, the peak summer temperature in our streams has been right around mid-August is when it kind of peaks. That makes sense. That's when the water levels are the lowest. It's been dry for quite a time. Temperatures are pretty high. That makes sense. But in 2021, with the heat dome we had, in the beginning of July, it peaked with our warmest stream temperature of the year. So that's very anomalous. But kind of the modeled climate change impacts in our area are showing that more of those kind of years are going to be more common and more severe. So if we kind of treat those as a glimpse into the future, with all of this years of data, we can identify those years and try to account for them being more common. And we can expand our restoration horizons, you could say, instead of just focusing on areas that, right now we know need the help, and the areas that are going to benefit most from the current conditions. We can kind of project forward and saying, okay, if more of our years are going to look like 2021, what other parts of our watersheds could also use work in that kind of context? So it opens up a lot of our streams that right now are like, they're not necessarily a high priority. They're doing fine. But if those years are going to become more common, they might actually need some work to be able to resist these climate change impacts, be more resilient to their effects. So when we have kind of 10 years of data, we can look at these bigger picture questions that you just simply can't do with 2 years of data. And that's why it's really exciting that we have this long-running monitoring program, and it's still going to be continuing into the future. So that's really interesting. So that was all of the topics that we had on that outline. We briefly looked at the motivations behind our monitoring, why we need to monitor and can't just piggyback off of someone else's work. We talked about the actual methods we use, the data we collect, why we use those methods. And we kind of finished there looking at some of the common uses of our monitoring data. So hopefully you learned something interesting and weren't too bored by all of that, especially some of the technical stuff at the end there. It's kind of a little dense. I'm kind of starting to really understand it after like months and months of looking at it, so I don't blame you if you didn't get it. If you are interested in this and want to kind of play around with it more, I would highly encourage you to look at the Luckiamute Watershed Council's data visualization website. The URL's there, and there's a link to it on our website, so you can just find it there if you want. This is a little screenshot from the website. You can choose which stations around our watershed and which years of data you want to look at. You can compare it to weather data for our watershed, kind of do all this comparison yourself and look at the patterns for the areas you are most interested in. Both the Luckiamute Watershed Council and the Marys River Watershed Council, we upload all of our data that we collect to the Oregon Department of Environmental Quality's Volunteer Water Quality Monitoring database. That's publicly available, so you can go download all the data we collect if you want and do whatever you want to do with it. It's out there. And all the funding for all of the work you've seen in this presentation has come through the Oregon Watershed Enhancement Board, OWEB, and they get their funding from lottery funds and from the sale of the state salmon license plate. So if you have played the lottery or bought the salmon license plate, you've helped fund some waterproof loggers and fund some very stylish waders I've been wearing out there in the streams. And I really want to thank again all of the volunteers who help with our data collection and the data analysis parts, and all the landowners who are involved in our program. Like I said before, it really makes our program far more powerful than it otherwise would be without all of their involvement. Yeah, that's a great question. So we're talking about how we actually take the sample. Is it just like a water sample, or what do we actually do? And then what species are we looking for in that sample? So we don't actually take a water sample. It's a kind of interesting method, so I'll explain it really quickly. It's developed by the BLM, who developed this procedure, and it works really well. So we don't actually take a water sample, but what we do is we have a pump and a little paper filter, and we pump 5 liters of stream water through a filter, and then take that filter and send it to the lab. And any little particles that are caught in that filter, that's what they search for DNA. So you don't actually need to transport water around. You have a little 2-gram paper filter that's really easy to mail off to the lab, and it works very well. And then when you do the analysis, I didn't mention this, but you have to specify which species you want the lab to look for, because there's so many species out there. They're not going to check every single species against every single piece of DNA they find. So you say, I want you to search this sample for coho salmon, steelhead, mussels, whatever, and then tell us if you find any of those in there. The species we look for mostly are coho salmon, steelhead. Those are always number 1. We work with the BLM, who helps us do this work, and they are very interested in Pacific lamprey, so they look for lamprey as well. And then we check for both the species of freshwater mussels in our watershed, the western pearl shell and the western ridge mussel. And then beyond that, it's kind of depending on the site-specific location. So if we anticipate there being western pond turtles there, we'll test for western pond turtles. Sometimes if we are interested if there's beaver in the area, we'll check for beaver as well. And that's about the extent of what we do, but sometimes there's other site-specific species that Yeah, that's a great question. That's actually yeah. So the question is, how far does the DNA travel down the stream? Like, if you sample from this location, where are you guessing the species, the animal could be? Like, where could it be? That's something that's really interesting about freshwater systems, is because you can do eDNA sampling just for like deer on plants. You can get like a sample of a plant and say, did a deer pass by here? But that's really difficult to do because the deer has to like touch that plant. But in a stream system, it's basically like a one-way path. The fish, or whatever we're testing for, has to be upstream of here. It can't be downstream because the DNA would have gone the other way. So it kind of limits the area. And that kind of question of how far upstream can you assume this species is, it's kind of an open question in science right now. It's kind of safe to assume it's probably within half a mile. That's like, okay, if you tested it, it's probably within half a mile. Or if you find the DNA there probably is that species located within half a mile. It might also be higher up. It kind of depends. It's past that point, it's not so sure. And some people who do the sampling kind of trust, hey, if we detected at this point of the stream, we're assuming it's all the way up the rest of the stream. That kind of makes sense, because if the fish got into this part of the stream, they can probably get up until the first blockage that they can't get past. So that's pretty safe to assume. But we generally stick with half a mile is what we use. Yeah. So yeah, I talked about how the log structures have more hyporheic flow and can help cool down the water. And you're right, when you kind of pond the water behind them, there can also be warming. So it's kind of like a dynamic system. You're not necessarily sure what the effects are going to be. And that's why generally we don't do log structures like that primarily just to address temperature, because they're not quite guaranteed to do that. We do those structures to accumulate gravel for other reasons, for spawning gravels and that kind of stuff. If they do end up cooling it, that's a benefit. It might do that. But I brought that up because a lot of people who kind of think about this assume that, well, you're kind of backing up the water and cooling it, so it must be warming. But that's not necessarily the case either. It could cool or warm. A variety of outcomes could happen. Yeah. So So the question is, we only have data for over 10 years, but do we see any results of the actual effects of our restoration work? Can we see that in the data? And the answer is right now, not really, which is kind of like we're doing something wrong. Like, why can't we see these results? But it really comes down to the time scale of these effects, right? If we plant a tree, when is that tree going to be tall enough to start having a noticeable impact on the stream? Not within 5 years. It's going to probably be 10 years before it even starts shading the stream, and 20 years before it starts having significant impacts. So in terms of temperature, where this kind of riparian planting is the dominant influence on because we talked about it with the log structures don't necessarily cool the streams by a lot, but they do accumulate lots of gravel, and they do it much quicker than trees grow. So we can actually see the effects of log structures on gravel accumulation pretty well. We actually have very good results from that, and we have data showing that. And we can see that after 1 year or 2 years, we start seeing some gravel accumulation. But this kind of long-term temperature effects, it's really tough to see because you have to picture all those different warming factors, and they all play into this area. So if we plant trees right here, that's going to limit the solar radiation hitting the stream here. But the temperature in the stream really is the accumulation of all the warming that is occurring here and all of upstream. So a lot of times the restoration work, if we're trying to cool the streams, it's going to have an influence downstream mostly, because that's where the water is moving after it's been warmed or cooled or whatever is going on in the system. So to get back to what your question is specifically, our data does kind of show that, but with only 10 years of data, and a lot of those years not really being after a project. They were kind of before the project in a lot of our cases. We're not confident of the scale of those effects yet. But maybe with more years of data, we can kind of get a better conclusion of that. But yeah, so the question is, a lot of these riparian plantings will also use water from the stream. They need water to grow. And how does that... Do we see the effect in our data, and how does that factor into our plantings? I would not be the best one to answer that because I don't really necessarily plan the riparian plantings as much. I don't know how much Aubrey takes that into account. But from what we see, we don't necessarily collect direct stream flow data at our sites. Actually, we do that in the Marys, but not in the Luckiamute. But the Marys, we only did it last summer for the first time. So we don't have enough summers of data to make conclusions about that yet. But at least in the Luckiamute, just from observations, we don't see a significant change. We're measuring depth, and from depth, we don't really see a drop in water levels from these areas. And kind of a lot of the idea is in these areas where we're planting these trees, we're often planting native trees, and the alternative would be blackberries or invasive trees that also use water, just they don't provide the same ecological benefits as Doug fir or other trees that we're planting. So the effect might not be as large as you would expect just from saying we're planting trees, but that is a concern. And in some of our streams where the problem, like we looked at one of them where the amount of water in the stream was the primary driving effect. In those areas, maybe planting trees like in the surrounding areas might not actually have as a benefit as kind of reducing the water draw. That could definitely be the case. We definitely need more data to be sure about that, but yeah, that's an important point to bring up. So the question is, are there any surprises in our temperature data compared to our eDNA data about where, temperatures compared to where species should be? That's definitely the case. Sometimes that comes down to the question of eDNA is great and it's very cheap compared to the alternatives, but the one downside is sometimes you can get a false negative, right? You take a sample and there just happens to not be the DNA of the species in that sample you took, right? It's not necessarily super likely, but sometimes that happens. And so you're like, okay, this sample we just took, it said there was no coho in the stream. But like last summer, we saw a coho in the stream. So like, is it just not there this year? Or did our sample come back as a false negative? So we have some of those where it's like, okay, we are pretty sure they're in this stream, but we didn't get that. So that's a surprise. But there also are some areas that I would consider, hey, it looks like great habitat, nice cool temperatures, great gravels, and yet like all the mussels, coho, and steelhead, and Pacific lamprey all come up negative. It's like, okay, that's probably not just a coincidence that all of that came back negative. And so there are those areas. And in those areas, we're thinking like, okay, what are the other factors that could prevent them from getting here? Are there problems downstream? Are there too small culverts? Are there problems, like are there really warm spots that the fish just won't even travel through to get to these areas? Like what else could be affecting that? And so that kind of leads into more specific questions for specific streams, and we can't like address that all in whole, but yeah, that's a very interesting point, so I talked briefly about us looking for beaver in the watershed through some of the eDNA work, and is there ever a case where beaver would create a problem? It depends. From like an ecological perspective, not really, but that's not the only perspective. A lot of landowners might have problem with beaver on their property. They essentially cause flooding, and that's what they're trying to do. And from an ecological perspective, that's what we want them to do. But flooding in the wrong areas negatively affects other people. If someone has, a road crossing and a beaver comes and blocks their culvert, and now they're flooding their road and washing that out, that's a pretty major problem. So there are problems with beavers, but from an ecological perspective, there not really are any. And you might think of, okay, we were talking about log structures and blocking up water and causing potential warming from that. Well, wouldn't beaver do the same thing, right? Beavers are going to be backing up water, more surface area. They tend to make large, wide, not very deep beaver ponds. So that would have a lot of warming. But it ends up that all of the other ecological benefits of beaver far outweigh the slight potential warming that comes from their ponds. They provide so much benefits to the ecosystem that slight warming is basically negligible compared to all their benefits. Hopefully that answered what you were looking for. So that's a great question. So these little monitoring devices, do we have problems with people taking them? How do we leave them there? How do we find them again after we put them out there? That's a very good question. So in terms of how we place them in the stream, we put them in little PVC casings, so like rocks rolling downstream don't crush them and break them, so they're kind of protected from the environment. We attach them to a 5-pound weight and a rope, sink them to the bottom of the stream, and attach the rope to a tree. So they shouldn't go anywhere, like they should be there. So if we come back to this spot, they should still be there when we come back. And then also we use camo rope, so hikers don't like see them in the stream. Like, what is that rope tied to this tree? Like, pull it out. But even with all of that, they do get taken sometimes. We have one in Rittner Creek Park, and it's been taken 2 times already where you had to replace it. I mean, it kind of makes sense. You walk along and you're like, what is this thing in the water? It looks kind of like trash. Like it's a PVC pipe and a weight in the stream. You know, you pick it out of the water. There's not. We write Luckiamute Watershed Council on it and the name of the site, but we don't necessarily want too many identifying markers. These things, despite being very useful and a self-contained unit, they're only like $150. So it's not like if one gets lost, it's game over. You know, like we can replace these. So yeah, that is fine, but that does happen from time to time. And in terms of finding them, that's very important. But we have a pretty good system down. If we're on someone's private property and we don't expect anyone to be like randomly coming across it and taking it out of the stream, you know, the property owners know we have it out there, so they're not going to mess with it. We will use flagging to really identify them so we can see this area, and we take very good GPS points of all of our stations, so we can go right to the point using our GPS, and it should be right around here somewhere. Oh, there's the flagging. Here it is. So besides people taking them sometimes, we don't really have too much problem locating them after you deploy them. But yeah, that's a great question. Yeah, exactly. So the question is, that model we were looking at with all the different warming effectors, is that statistically based with like just looking at the relationship between these factors, or is it mechanistically based? It's all statistically based. So you're right. And so because of that, we can't directly say that this amount of discharge caused this warming. We can just say, you know, the amount of warming is most closely associated with the amount of stream flow in the stream. So in this kind of presentation where we're not necessarily using the results here, I kind of simplified over that. But in the kind of written report I have, it's very clear about this isn't showing this causes this. It is just kind of a relationship between them that we've noticed, basically. But yeah, you're right. That's an important point to mention. Yeah, so the question is, we're not doing any monitoring in the Ash Creek system right now, and why is that? You're right. There's kind of this, at least in the Luckiamute, you know, I kind of mentioned this a little bit, how the Luckiamute historically has done all of our monitoring in the headwater reaches, where salmon spawning is most likely to occur, and there isn't really any salmon spawning in Ash Creek. So that's kind of why historically there hasn't been monitoring there. But we are actually looking now at trying to add stuff there. Kind of the Teal Creek Little Luckiamute area is our first expansion, because that kind of fits more with what we have been monitoring, and it's a really high priority. But this summer, on that eDNA map, I didn't have the points that we proposed to sample this summer, but we're proposed to take a lot of eDNA samples in Ash Creek to see. Because Ash Creek also has the benefit of potentially Chinook salmon are using it for habitat coming off the main stem Willamette. And so if any of you don't know, Ash Creek doesn't flow into the Luckiamute, it just flows straight into the Willamette River. So while it's not part of the Luckiamute watershed, we manage it as a watershed council and do work in that area. Yeah, so from the temperature perspective, it isn't as high priority as the other parts of the watershed, but from an eDNA perspective, it is, and we're hoping to try to address that problem going forward. Yeah.